Welcome back. Next, we have Bioxytran Inc. They're an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug, an oxygenation molecule being developed to treat stroke, and its development is supported by an FDA-approved device that detects local tissue oxygenation. So due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. And the company has a third platform technology that is capable of treating cancer metastasis. I always say that wrong. Mike, you can explain this better than anyone. It's always good to see you. And I know that at the last conference, you gave a teaser of your cancer metastasis presentation. But today, I understand you want to kind of talk about the significance of your last peer-reviewed journal article on viruses and the significance of it. And in your press release, you heralded your journal article as a medical breakthrough in fighting all viruses. Wow, that's a bold statement. So what I'm hoping you can do is back this up and give investors your take on what this means. Welcome back, Mike.
Well, first of all, I want to say thank you for hosting this conference, and I want to thank all the investors listening in on this call. What we have on our hands is, in fact, a major medical breakthrough. I mean, we knew we had something back in 2021 when we saw 100% efficacy in our phase 2 trial. And we technically weren't even checking for efficacy at this point. This peer-reviewed journal article, in essence, captured our drug development methodology. But here's the thing, we did things in reverse. First, we found out in our clinical trial that the galectin blocker we made was non-toxic and had profound efficacy. Then we did in vitro studies on COVID-19 and found out that it could neutralize the virus. But we weren't totally sure if the mechanism of action, so we did an NMR study to assess the binding affinity of our molecule to the spike protein. This is where we validated our hypothesis from way back in 2020, where we believed we could neutralize the virus by binding to the spike proteins and just interfering with its entry. Now, this methodology left us with the formula to design new antivirals capable of treating practically any virus. After we pick the virus that we want to target, sorry about that. After we pick the virus, where was I? After we pick the virus we want to target, we can basically use nuclear magnetic resonance imaging to find out if the spike protein of the virus binds to our antiviral. The beauty of these NMR tests is that they not only tell us if the two molecules bind, but how strong the bond is. Mathematical algorithms combined with artificial intelligence software are used to plot where the binding regions are in space, as well as their binding strength. Now, once we find a match with these NMR screening, we can run in vitro tests to see if we can actually neutralize the virus. We can do all this development work before we even enter human trials. Once a drug's in human trials, there's a very high likelihood based on this methodology that the drug's going to work. Now, we have one patent pending on lectin-binding carbohydrates to treat viral infections. Now, this is a very broad patent and covers a multitude of drug formulations that could treat well over 60 viruses. Now, I want to invite investors to look at our press release from way back in last year, this time, because we think we have solutions to some of the most lethal viruses, like Ebola, Marsburg, or Nipah virus. Now, let me shift gears and talk about the actual discovery that we made. The discovery relates to how viruses infect our cells. Now, the existing theories paint this picture that the viral spike proteins attach to cell surface receptors and they trigger cellular response and allows the intake of the virus. Our discovery reshapes what we know about this viral entry. We found out that galectins actually help facilitate the viral entry by acting as this like little tethering mechanism between the cell surface receptor and the viral spike protein. We discovered a conserved binding region on the spike proteins on a number of viruses, which enables the capture of the viruses. Now, think of galectins as a viral magnet that attracts the virus to the cell. So the good news is that we discovered the Achilles heel of viruses. But the bad news is that no one in virology really understands what we're talking about, or heck, they be throwing money at us. There's a major disconnect in what we've discovered in virology in general. Almost all antivirals work by stopping or slowing the production of viruses once in the cell. We attack the problem from an entirely different direction and think it's smart to actually keep the virus quarantined outside the cell and preventing the viral entry. We're on a completely different level with respect to the rest of virology, which is why no one really gets us. I mean, the truth is, we could be sitting on a cure for HIV, but we won't know until we get more funding so we can do a battery of testing. I hope investors realize the explosive upside, considering we're about in 1,000 on our in vitro studies, and we think we can keep this stroke going.
Well, Mike, let's assume for a moment here that your antiviral technology has the power to block a number of viruses. Could this mean the end of the jab as we know it?
No, we all hope. Right? Actually, vaccines do, in fact, serve a purpose. Vaccines like measles, mumps, rubella, you've all heard of these things. What about tetanus or rabies shot? These are what we call sterilizing vaccines, which means if we're exposed to the virus, we're not expected to get sick. Now, I expect these type of sterilizing vaccines, they're going to stay with us for some time. There's no reason to sideline them. The real issue is with the current vaccination strategy. And its definition of a vaccine has changed from preventing any form of disease, like the sterilization, to what we have now, which is get sick, but get sick with the lesser form of the disease and avoid hospitalization and death. Now, the trouble with vaccines, in these type of vaccines, is you need large buy-in from a sizable part of the population for it to be effective. Now, during the pandemic, the magic number was around 70%, but the uptake this year is under 15. That's just not going to work. People are realizing that vaccines can't keep up with the mutations. And then the side effects of the vaccine, they're basically on par with just getting sick with COVID. So the mindset is, why bother? Now, Bioxytran's antiviral platform could make the seasonal vaccines referred to as the jab obsolete. It makes no sense anymore to vaccinate on such a large scale when a therapeutic drug exists that's capable of treating the disease very quickly with no known side effects. Prolactin M demonstrated 88% efficacy in 3 days versus 0% in the control in its phase 2 trial. I want investors to remember that. The company believes with adequate funding, we can develop antivirals to basically treat almost any virus.
Well, I certainly hope you get funding. I hate catching colds. I'm sure most of our viewers do too. And I know a lot of people don't want to get the jab. So when I catch a cold, I feel lousy right away and then I get over it. But I hear cancer obviously is a very different animal because you start feeling off, but then it gradually gets worse and worse. So what's the linkage between viruses and cancer? Because these sicknesses behave differently.
Okay, when you catch a virus, you get sick pretty fast because it takes a while for your immune system to kind of kick into gear. So the most successful viruses are going to find some way to shut down and slow your immune response. It just kind of makes sense, right? The same thing happens in cancer. The immune system gets turned down so the cancer has a chance to gain a foothold. Once that cancer's defense mechanism kicks in, it just kind of finds a way to make itself invisible to the immune system. So the question you may have is how does this happen in a viral infection and in cancer? And I got a slide for that. So here we are with our forward-looking statements, and here is the slide. So in this, let me set the stage before I talk about this slide. Let's put ourselves into Star Trek mode and visualize a solar system. Pretend the planets are healthy cells, and the immune cells, which are referred to as T cells, these are big space stations that travel between planets and check to make sure there's no aliens living there. Now, the aliens could be viral colonists or cancer colonists, but it really doesn't matter because our T cell space station is equipped to destroy either one of them. Most of the time, the space station checks each planet and finds no evidence of alien life. But let's say it did find an alien colony. It's going to trigger a response where it's going to move into the very low orbit and open a massive, shiny, curved solar blanket that covers half the planet. And it's kind of like a magnifying glass concentrates light. This solar blanket would focus this death ray to the planet, killing all the aliens. Well, this is how T cells normally patrol and get rid of alien invaders. Now, there's a great video that shows how T cell works, and I'd like to play this for us now. It's 1 minute of our time, and then we'll move on. Please play the video.
Inside all of us lurks a serial killer. A killer whose primary function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells. And they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
Okay, so I hope that video really helps put things into perspective on how a T cell actually works. See, it gets that good adhesion. Now, let's get back to the analogy that I was talking about. Let's say one of these planets has viral colonists that like to mine the planet, and what they do is they launch this massive network of satellites, which we would call galectin 3, into low planetary orbit. So this time when the space station scans the planet and finds alien life, and it tries to move into lower orbit to focus its death ray, what happens is it can't do it because all these constellation of satellites, it's in their way. It's disrupting their ability to focus the beam. And this is both how cancer and viral infections slow down your immune response. So now I'm going to apply this to my slide deck here. All right. So you see a slide deck called T cell energy. The picture of the T cell on the first graphic on the right, that's how you get good adhesion. You see all that blue? It's getting good adhesion. It's sticking to the cell really well. And those blue things are little grappling hooks or lectins, and they're distributed throughout the surface of the cell, and they come and they coalesce at this point. And that's how it's supposed to work. And you saw that in the video. Well, what happens is this constellation of galectin plaque gets thrown up at the T cell right as it enters, as it enters to dock on the cancer cell, and that's the graphic that you see on the left rather. So what happens is these lectins they can't get good adhesion. All right. So that's the thing that I want to explain about this. The next slide that we have here, this is how this effect happens not only in viruses but also in cancer, and it's called T cell anergy. And but what happens in a viral infection is galectin-3 is upregulated. So it's a protein that the virus used to make the outer shell of the virus capsid, right? And what happens is it gets upregulated, and then the cell runs out of nuclear material and it destroys itself. It pops, and then all this galectin-3 materials in the way. And what happens is that's what ends up affecting your immune system in viruses, and that's why it turns it down. This slide it goes over the video that we just showed, and this shows the impact in cancer. But I'm pretty much done with my slide deck, so now we're going to get back to your questions.
Okay. Great. Well, Mike, you've come onto the Emerging Growth Conference a number of times, and you've talked about viruses, cancer, fibrosis, stroke, and neurological diseases, a wide range of topics. So what's the common thread between all these diseases?
Inflation, not inflation, inflammation. Now, you see my side? Now, I want everyone to pay attention now, because you need to understand what Bioxytran is about. We are about developing drugs for people that basically takes inflammation out of their lives forever. And I want people to grasp the magnitude of this medical discovery and how it's going to change the world as we know it. Now, this slide I call the galectin troublemaker slide. And what you'll see here is this inflammation cycle. And what's at the center of it? It's galectin-3. It's right at the center of this disease. And I call galectin-3, if you saw my first slide, the apex predator of inflammation. It's the top of the food chain when it comes to inflammation. So the bottom line, you block it, and this whole cycle dies down. And you see there's these little double arrows. Those are negative feedback loops. And we can get rid of that. So anyhow, that's pretty much that's it for my slides. You have some more questions?
Sorry, I wasn't sure when you were done with that. If you can give our audience an update on any of your licensing initiatives with big or small pharma.
Yeah, we're finishing up a very extensive cancer slide deck in anticipation of positive clinical trial results expect to be released in December using a galectin-3 antagonist. I want to be clear, though, that these trial results that we're waiting for, they're not ours, but we believe they're going to generate a lot of interest in the sector. So we want to be prepared. Our domestic focus is on dermatological applications and virology, but we're also testing the water with some prospects regarding use of galectin antagonists in cancer. Now, I'm also very pleased to announce that we just hired an international specialist who focuses on big pharma in Japan. And he's already confirmed 3 in-person meetings for next month. We have some very good, he has some very good referrals and used to do a lot of work with Vertex Pharma in their early gestational phases of development. The nice thing about him is that we have a success fee driven contract with him. And our goal is to get signed NDAs as soon as this month. So we can basically get started on licensing initiatives ahead of this December readout that we're talking about. Now, one of the strengths that he saw in our pipeline is our versatility. He explained that a majority of his client base basically needs a broad inflammatory type of drug. And galectin blockers attack the heart and soul of that cycle of inflammation. Remember the galectin-3 apex predator?
Yes, I do. I do remember that. And you mentioned you're waiting for some clinical trial results to come out in December from another company. And I think you also talked about that at our last conference. So is there any chance you can tell investors what company you're mentioning?
Good one, Anna. Well, I typically shy away from promoting my competition, but I'll give investors a clue if they really want to know the answer. This is where I get to do a little self-promotion. The answer to your question is in my LinkedIn article, somewhere between the beginning and the end. So you got to read the whole article, and don't forget to subscribe, like, and share. You can find the link in our Twitter feed.
All right. So in your article, it seems like you bring up the ethics of dosing patients with Keytruda, who clearly aren't going to respond because they have this galectin-3 in their bodies. So is there someone we should blame for this? Don't drug companies have a moral obligation to put their patients first?
Well, you got to read the article, and I invite everyone to read the article. I really think there's a moral, if there is a moral obligation, it's going to reside in the hands of the oncologists. It's basically at the oncologist level. These are the frontline responders in the war on cancer. And I'm not sure you can really point blame either. Is the oncologist's fault that they didn't read a journal article from 5 years ago that basically says, you know, the galectin-3 expression level from a tumor biopsy, you need to do this before you even think about prescribing Keytruda. You know, this isn't the oncologist's job to research other tests that could guide their treatment options. Now, about 8 years ago, the FDA and Merck decided to do a PD-L1 test that would decide the eligibility criteria needed in order to prescribe Keytruda. This is an FDA-approved test, so it's highly unlikely that a drug company could interfere, let alone want to interfere. So just so you understand the process, let's just talk hypothetically. Let's assume that oncologists stopped ordering Keytruda out of the blue. I guarantee you Merck was going to want to know why. And they're going to investigate and find out that, oh, oncologists were just running this additional galectin-3 staining test before they administered Keytruda. Oh, that's interesting. So in this type of scenario, you're going to have basically well-versed oncologists felt they, quote, had what you described earlier as this moral obligation to put the patients first. So at this point in the process, Merck is going to commission, in this hypothetical process, Merck is going to basically commission a clinical trial study with, and basically test the oncologist test that they're using, this galectin stain that I talked about earlier. And they'll work on getting it validated and ultimately approved by the FDA. Unfortunately, there's no real mechanism in place to address a situation like this except market forces. Now, let's go back to the hypothetical. If a drug company starts losing sales, they're highly motivated to run a clinical trial so they can make a claim like, hey, if you take galectin-3 test, there's a 100% chance you're going to respond. I mean, that's kind of a cool thing to say, and it's like the ultimate marketing gimmick. Anyhow, so it's the ultimate marketing gimmick. Now, I have to give you one little caveat here. It's about the status quo, and which we have right now, because there's two independent studies that explain Keytruda resistance, and both of them are analyzed in my LinkedIn article. So here's another plug, read the article. Now, all the PD-1 inhibitors have neutralization concerns, but I'm most concerned about Keytruda because the data indicates the drug could be completely neutralized depending on the level of galectin-3 concentration. What I'm finding hard to reconcile is whether or not this should be elevated to concern of patient safety. So on one hand, these were independent studies, but if the drug company did these studies, they would be hard-pressed to withhold this from the regulators. Now, my gut tells me that this has turned into a patient safety issue. Medical ethics revolve around this core idea of doing no harm. And Keytruda does, in fact, have side effects. And the level of galectin concentration is correlated to neutralization of Keytruda. So there's a viable argument that Keytruda may cause harm if the galectin level isn't known. Now, I'm not advocating pulling Keytruda and Tecentriq off the shelves, because that's going to hurt a lot of people, and that would do more harm. But I think this issue has been swept on the rug for way too long, and that the regulators should be involved. This is tantamount to giving a terminal patient who has cancer a placebo. You just don't do that. But what I think is going to happen is basically in this little gestational phase right now. See, Merck's the maker of Keytruda. They're under a tremendous amount of pressure to make their drug better. Summit Therapeutics just came out with a competing PD-1 inhibitor, just like Keytruda, but with dual action that's also going to stop the blood supply to the tumor. So Merck can't sit idly by and just collect this dividend off of Keytruda. The competition is forcing them to make improvements, and it's not a huge leap for them to start connecting the dots and realize that galectins neutralize Keytruda, and galectin blockers could reverse that effect. Now, it's also worth noting that the ultimate knowledge of making a galectin inhibitor, it's in our company. Investors need to know that. And we actually have the person who coined the name galectin and developed the largest number of galectin antagonists that are out there. So our company is actually best suited to develop any solutions that will require a galectin antagonist.
And what about this drug, ivonescimab, from Summit Therapeutics? I can't believe I said that correctly. That drug supposedly had better results than Keytruda. Thank you. So in a head-to-head study, better results. So do you think galectins will interfere with their PD-1 blocker like it did with Keytruda and Tecentriq?
Yeah, they had really good results. Now, there's no way to tell for sure without a head-to-head comparison. That's why those two went head-to-head, and ivonescimab won. So here's the thing. The other PD-1 inhibitors were interfered with galectin-3. So it's very possible that trend's going to continue. So regardless of what PD-1 inhibitors use, it's important to understand that we believe a galectin-3 inhibitor is basically going to fix the problem.
So let's get this straight. You have 100% responder rates in 2 COVID clinical trials. You have in vitro tests that show your galectin blockers also neutralize RSV and influenza. You wrote one article detailing why you think your galectin blocker has the potential to treat bird flu. Now you have 2 articles on why you think this will work in cancer. So how much more scientific proof do you think you need to put out there before you think investors will pay attention and buy up the stock?
Truth is, I think HIV, I think that's our ticket. There is so much data out there that says galectin-1 and galectin-3 play a major role in the entry process of HIV that this has to be our priority when we even get a small amount of funding. We need to find out if we can neutralize this damn virus once and for all. Investors don't seem to care about treating COVID-19 anymore. But when it comes to HIV, they're a little bit more responsive and passionate about making this virus extinct. So, bottom line, I think once we finish our HIV tests, that's going to be the tipping point.
Wonderful. Well, Mike, as always, a thorough, fascinating conversation with you. What are your closing remarks for our viewers today?
Well, I want to thank everyone for being here. I want to encourage everyone to read my latest article on Keytruda, the third wheel. Check it out. It's on my LinkedIn profile. I think it's going to enlighten you to the real possibilities that we have here. You know, we have the potential to make something that's that's a $25 billion drug better. What's that worth? Well, investors, you be the judge. Thank you.
Wonderful. Thank you, Mike. Nice speaking with you. And I look forward to our next conversation.
Sounds good. We'll be here.
All right, everyone. See you in just a second. We'll be right back.